So this is our model of the light reactions in photosynthesis. So this is the thylakoid membrane. This is the inside of the thylakoid. This is the outside of the thylakoid. Step one is photon hits the chlorophyll, which is a light-absorbing pigment, in photosystem 2, which is right here. And the energy from the photon excites the electron into a higher energy. And then this electron carrier carries the electron into the cytochrome complex. And while this is happening, H2O splits and supplies photosystem 2 with another electron, and O2 is a byproduct. And the excited electron in the cytochrome complex, the cytochrome complex uses some of the excited electron's energy to transfer protons into the thylakoid lumen, which is in the inside of the thylakoid. And that's it for my part.
What's up, guys? This is Patrick. I'm picking up where Micah left off. Um, Micah left off right here, where an additional electron carrier carries this electron to the cytochrome complex, and then the cytochrome complex sends it to the PS1, where inside the PS1, a photon excites the electron, so it has enough energy to be sent up to an enzyme with hydrogen and NADPH or NAD P positive to help form NADPH. The reason why we need NADPH is because NADPH is a reducing agent produced by the light reactions of photosynthesis.
So before I get any further, I will be talking about what a thylakoid membrane is and what its whole role in photosynthesis. So here we can see a model of the thylakoid membrane with all the different labels. So a thylakoid membrane is a highly folded internal membrane system found within chloroplast of a plant cells, where it acts as the site for the light-dependent reactions of photosynthesis. So basically everything we covered before, this is basically where everything occurs. So picking up where we left off, protons accumulate in the thylakoid space during the electron transport between photostem 1 and photostem 2. And then they fuse into the stroma through an enzyme called ATP synthase. A recap on the stroma is it's the cells and tissues that support and give structure to organs, glands, or other tissues in the body. During this, ATP synthase uses the potential energy of the proton gradient to combine ADP with inorganic phosphate, which creates ATP, which mini-me's kind of pointing at right now. So again, this is why this buildup of inorganic phosphate is so important, as kind of one of the key factors as to why and how ATP is produced. So after all of this is said and done, NADPH and ATP have stored energy from the light reactions and can now be used for the dark reactions or the Calvin cycle, which I'll recap a little bit about. So again, as mentioned before, from PS1 to PS2, ATP is created via the protons going down the proton gradient that's made from all electrons going down the ETC, which is just an electron transport chain. The ATP from this is taken to the Calvin cycle for later use. It all starts with the 6-RUBP, which combines with the CO2 from the air to form PGA, which is a 3-carbon sugar. This moves down, and with the energy input of 12 ATP and ADPH, it forms PGAL. Where it then breaks 2 off to leave the Calvin cycle, and 10 PGAL stay behind to regenerate the RUBP with the help of the 6 ATP from before.